Welcome everybody to the Kennisclip Amelogenesis. My name is Sarah Pringle. I've given you the Kennisclips about embryogenesis and dentinogenesis, and now we're going to talk about how... Okay, what is enamel? So enamel is the hardest part of the, actually your whole body, not even just thinking about your teeth. It's harder than bone, and it's the outside covering of your teeth, so this is probably really familiar to you guys, the white layer on the outside. It does not contain any cells. And what we need to know, what you need to know for your development as future dentists, is how this is made. So amelogenesis, so we mentioned this in the past Kennisclips, is a process that happens in collaboration with that of dentinogenesis and is mediated by ameloblasts. So you can see here, this is a schematic which I will come back to through the course of this Kennisclip, running from stage 1 here, 2, 3, 4 and 5. So the cartoon is meant to show how you get to a stage when the cells are producing the precursors to enamel, so this kind of phase here, and the modulation of the enamel. If you remember back to your embryogenesis Kennisclip and knowledge, ameloblasts differentiate from the internal enamel epithelium. So again, the embryogenesis knowledge is important to you here, to understanding where these cells come from. It also begins at the enamel-dentine junction and happens in these gradual phases like you can see happening here. So let's go into more. Phase 1. So this phase in the red box on the left-hand side is the pre-secretory phase, is its name. What happens here? So as you can see here, the cells are still quite square, almost in shape, and they move into this elongated shape. To do this, the cells of the internal enamel epithelium, they stop proliferating because now they need to turn into functional ameloblasts. They become longer, columnar in shape, and the nucleus moves away from the dentine. So if you remember what I said in the last Kennisclip, dentinogenesis always happens first, and this is a signal which stimulates the ameloblasts to differentiate and to get ready to make the precursors for an enamel. So the nucleus moves away from dentine, like you can see here, so here would be the dentine underneath, and the secretory organelles are formed. So here they're in the picture like these yellow little shapes. Like I just said, odontoblasts have already started before with dentine production. The basal lamina, which normally in this image is here, this really low, thin black line here, basement membrane, proteins like laminins and fibronectins, is resorbed by the ameloblast. Resorption meaning disintegrated. So here you can see in the cartoon that it has holes in it, so it's being degraded. Where this happens will mark the position of the future dentine enamel junction. And as this signal, the presence of dentine, which is made by the odontoblast, provides a signal from the ameloblasts to degrade the basement membrane and to begin this process. So that's stage 1. Move forward to stage 2, the secretory phase. So as you can imagine, so here looking at pictures 3 and pictures 4, I've bundled them together because otherwise there are loads of stages. This is an action phase where you start to see production of proteins which will eventually be mineralized to make enamel. So this pre-enamel component, remember, epithelial cells, well, any cells, do not secrete hard crystallized forms of the final tissues of the tooth because they would die. They secrete a pre-enamel component. So you can see this represented here by this kind of dashed line is supposed to represent the pre-enamel component. The secreting pole is what it's called, this triangle shape here. This is called the Tomes process. It's very easy to recognize under the microscope, and it's a very distinctive part of the development of ameloblasts into their functional state. So the organelles move towards the Tomes processes and they begin to secrete. So what you can see here also, which I will come back to later, is that this Tomes process here is secreting the enamel precursor components in this kind of direction, but you also have secretion down the sides in another direction. So this will make more sense when I come back to it later. So that's phase 2, the secretory phase. If we focus more in on this phase here, which I was just talking about, what happens first, there's a difference between two different types of pre-enamel that are secreted. And in your books and online and everywhere you look, you will often see the words interprismatic and intraprismatic enamel stated. And that comes because of this difference, like I just mentioned, between enamel that is secreted down the sides, or the precursor proteins for enamel, and enamel which is secreted in the middle. In amelogenesis, this sideways secretion is actually what happens first. So here I've tried to represent it in terms of a whole panel of ameloblasts, so they're all lined up together. And the red represents the precursor protein secreted interprismatically to start with, so in between here, which, if you kind of imagine a whole layer of ameloblasts, means that next door ameloblasts, now looking from above, so this is the cell nuclei and this would be the cell body, these enamel bits that are secreted contribute to making it a kind of wall, like a tube of enamel between neighbouring cells. Hopefully this will look more familiar when we look under the microscope. When they have made this kind of wall, the ameloblast retreat behind. So you can kind of visualize it like this, I would think, that neighbouring ameloblasts collaborate together to secrete initially interprismatic pre-enamel proteins, making a tube which they then move away from, and then they proceed to fill the tubes, which is then called the intraprismatic enamel. I like to think of it like honey and the honeycomb. So now each of the ameloblasts secretes this portion here, so from the Tomes process directly into the empty tubes which were just made by the collaboration between its neighbours. I hope this makes sense. So now you're looking above from here. So underneath I wrote each empty interprismatic enamel tube, so you should really read that as pre-enamel tube, is filled by the secretions of a single ameloblast. So it should be filled in like this. And then moving into the last phases of amelogenesis, phase 3 to 5, so also 5 to 7 in this diagram, transition, maturation and post-maturation. So what do we mean by this? A substance which is made by cells, secreted, a precursor substance, is going to be way too soft to perform the function that enamel eventually needs to perform. So it needs to be adapted, it needs to be modified, dramatically modified. In order to do this, 95%, so that's the vast majority of the organic matrix which was secreted by the cells, needs to be absorbed. We need to remove the water, we need to remove the protein. So in order to do this, ameloblasts shift into this last stage of their life cycle, which is pretty amazing, that they can secrete something here in such a dramatic process and then shift to such a different cell morphology. In order to do this, you lose this Tomes process. So you can see there's no longer a triangle end of the cell here, like this. Now you have these kind of wiggles, which are called the either ruffled or smooth cell surface. So ruffled meaning all of these waves here, and smooth meaning this waves. In between these two phases are the phases of the ameloblast life cycle which perform the reabsorption of the water and protein. And the theory is that the reabsorption happens in this phase, this ruffled phase, and that this smoother phase here is is a resting phase, or sort of, between these two cycles, allowing the cells to recover from reabsorbing all of this organic material. So that less pumping and endocytosing of the organic matrix from the pre-enamel happens here. Here it's happening a lot. Also what happens in this process, and this is given by cell 7 here, I believe, up to 50% of the ameloblasts will die. So 50% undergo apoptosis. This final stage then you can think of as being split into two processes. So if you lump these guys together, this is the resorption of water and protein, shuffling between this ruffled phase and smooth phase, actively resorbing here and resting here, and dying through apoptosis here. If you put these all together, so now you need to think back to the previous Kennisclip about odontoblasts and dentinogenesis, ameloblasts and enamel. So this is how I remember them, with the D's and the AMs. On the top row of this figure you can see the life cycle of an ameloblast, more or less matched together with the life cycle of an odontoblast. So this should look familiar to you. Here is the differentiation from pre-odontoblasts into odontoblasts, the secretion of the pre-matrix versions of dentine here, and then the elongation of the processes of the odontoblasts, which are characteristic of dentine. You can also see here, so pink is the basal lamina, the membrane. And you can see when there starts to be dentine here, this stimulates the digestion of the basal lamina by the ameloblasts, so it's gone now, and the activity of the functioning of the ameloblasts in terms of producing the precursors of enamel. And here again are those two different directions of enamel precursor secretions. So bear in mind this is always a interplay between two different cell types that need each other to successfully form the hard tissues of the tooth. How does this look under the microscope? So this is something you are also expected to know. Remember I was saying that amelogenesis, so formation of enamel, happens at the enamel-dentin junction. So if we go back to one of our cartoons that we were focusing on before and zoom in on this part, so here is the dentin, we already talked about that. Here are the ameloblasts, which were differentiated from the inner enamel epithelium, and now we have an orange layer, which is supposed to represent the precursors of enamel. And if we zoom in, you can then quite easily see, I hope, this is the pre-dentin, hasn't been mineralized yet, it's just been secreted by the odontoblasts. This is the dentin, which has been mineralized. This is the styloid intermedium, and you also have these longer cells here are the ameloblasts. And then here you have the enamel, which is very colors in this staining very darkly. It's very easy to see. The beauty of looking at ameloblasts in terms of enamel generation is that you can really see them under the microscope because of this tomes process, this triangle structure that I was talking about. So again, if we zoom in, I hope you can see here, this dark black at the bottom will be the enamel proteins, pre-enamel components secreted by the ameloblasts. And you can quite clearly see this is the line between the cells, cell number 1, here's another cell, cell number 2, here's another one, number 3, and you can quite clearly see these triangular tomes processes, and which would be responsible for secreting the precursors of enamel, which fill in the tubes which are made by the initial secretion from between the tomes processes. I hope this is clear to you. One more time to hammer this message home. From interprismatic enamel and intraprismatic enamel, if we look under the electron microscope, so super high power microscopy, you can see very clearly, I think, these thin walls of enamel here would be the interprismatic enamel, which is made first, these tubes which are made, which is then filled by these larger areas of intraprismatic enamel, and each of these large areas of intraprismatic enamel is made by one ameloblast. These walls around the outside are made through the collaboration of several ameloblasts. How does this look like in terms of timeline of human tooth development? So you can see in terms of age of the embryo, So dentine starts to form around 18 weeks of development, and ameloblasts also, which means there is between this point and this point the appearance of enamel in the developing embryo. I hope I've been able to show you, to summarize today, 3 different things about amelogenesis. So looking at ameloblasts, the cells responsible for the production and secretion of the pre-proteins of enamel, how enamel looks like under the microscope, so thinking about inter and intraplasmatic enamel in this honeycomb format, and how you can recognize enamel when you look under a microscope in terms of H&E staining on this area of the tissue here, the enamel. Thank you very much for listening, and I hope this has been a relatively clear explanation of what can be quite a complex concept. So today I hope I have outlined for you the process of amelogenesis, looking at the ameloblasts, the cells which are responsible for making enamel, in the end, the different life cycle stages, how enamel looks like when you look at it under the electron microscope, so in terms of inter and intraprismatic enamel in this honeycomb format, and the microscopic appearance of enamel in terms of H&E staining, like you can see on the right hand side, and how you can recognize it in a developing tooth. So that was the end of everything I wanted to tell you today about amelogenesis. Next Kennisclip from me, number 4, will be about root formation, and then we talk about eruption and the gingival sulcus. Bye bye, until next time.